Good morning. Here we have the star of SIPS 2024, Professor Hershko, and it is 20 years of anniversary of being awarded Nobel Prize for his discovery. Professor Hershko, it is a real pleasure and honor to be with you again. How are you doing?
I am good, thank you, thank you.
So, how you feel in 20 years? 20 years anniversary of your Nobel Prize.
The years are passing. Yeah, years are passing quite quickly. I should say. Yeah. So it has been really 20 years since I got the Nobel Prize, together with my colleague at that time, Erwin Rose, and with my past student, Aaron Chekhanov. Yeah. So it was a very exciting event for the discovery of the ubiquitin system for protein degradation.
And what is interesting is that the rare professor in the world have a Nobel Prize with their student also.
Yes.
So it is. This is exceptional. This is really, really rewarding to have a Nobel Prize with a winner. There are a few examples in the world, but not too many. 20 years for your university, and there is this entire summit dedicated to you. It's a kind of people coming from all over the world, about 500 presentations from 65 countries in this summit dedicated entirely to you. This presentation will be showing in the opening of the plenary session. The entire plenary session will be opened by your presentation. So, Professor Hershko, can we start with your presentation? This is a very particular and very special presentation on the 20th year of the anniversary of Nobel Prize. It is quite an achievement, 20 years.
Yeah, thank you very much for inviting me to this important SIPS conference and for honoring me by dedicating this meeting to my honor. So it's a really great honor indeed, and I am very thankful for it. Unfortunately, I could not be present physically in that meeting, but I would like to send this video recorded lecture to be presented with some of my thoughts about the importance of basic research for industry, for innovation in industry. So why I think that basic research is important? It's important for industry because most of you are working in applied research. My background is medicine. I studied medicine about 50 years ago, and then I got interested in biomedical research, and I went on to research. When I compare medicine as it is now to what it was 50 years ago, there are some huge changes. Can I have the first slide, please?
Sure.
So here is some of the changes. During these 50 years, there were very great improvements. So there was a huge increase in life expectancy. In most Western countries, life expectancy went up by about 10 years during these 50 years, which is a huge increase from about 70 or more to 80 or more years of age, life expectancy. And this was due to some great improvements in medicine. Some of the improvements were in the prevention of disease, and that's very important to prevent disease. For example, cardiovascular disease is now much better prevented than 50 years ago by the use of statins, for example, that lower cholesterol, by management of hypertension, and very important by more healthy lifestyle, the use of exercise, diets, and so on. So all these things added a lot to the prevention of cardiovascular disease, and there was a great decrease in the incidence of cardiovascular disease. There were also great improvements in the diagnosis and early detection of disease. For example, now we have imaging such as CT, computerized tomography, MRI, or ultrasound technologies. All these are now commonplace technologies today. They were not there 50 years ago. So the great one of the revolutions, or the great revolutions in medical care, is actually due to technological revolutions of better imaging and better medical instrumentation. There were also great improvements in technology, such as in such as in such as in intensive care, pacemakers, and all that added a lot to the better management of disease, and to great improvement in medical care during the past 50 years. Now, all these improvements were due to research, to research, and the type of research that are known are shown in the next slide.
Yes.
Yeah, so there are basic research and applied research. In basic research, we want to know how nature works, how things are working. So it is led by curiosity. We just want to know things. And because we want to know, we make discoveries. But basic research is not just to satisfy a curiosity. Basic research is also used then later for applications. So you cannot have applied research directly improves care, medical care, for example, or it improves industry. But you cannot have applied research without basic research. You cannot have, for example, engineering without great discoveries in physics. That's clear to all of you. So you cannot have medical improvements in medical care without basic research. So the basic research is the basis of applied research. And I would like to show some examples that prove this point. So here you see Watson and Crick they discovered the structure of DNA right after, with their model, right after their discovery. So they were led by pure basic research related curiosity. They wanted to know what is the molecular basis of heredity, and they found that how DNA is arranged by the double helix, and this arrangement explained heritage. So they were led, as far as I know, by basic research. They never thought of application. However, their discovery caused huge ways of application. Now you know about precision medicine, in which medicine is applied to individual people, as it is done by DNA sequencing. So by sequencing the DNA, by identifying the sequence of certain genes, we can now direct medicine to individual patients. You also have recombinant use of recombinant proteins, for example, humanized insulin, or you have all these monoclonal antibodies that are made to treat a large number of diseases, including inflammation, cancer. All these are wonderful diseases, wonderful agents, recombinant agents, and all these are based on the discovery of Watson and Crick of the DNA structure and the genetic code, and so on. DNA sequencing is even used, as you know, in criminal investigations. So they had a huge impact that they didn't think of when they made the basic discovery. But basic discovery was the basis of all these. Another example is shown in the next slide. Here is Alexander Fleming, a British microbiologist who discovered penicillin. And he discovered penicillin by an accidental observation. One day, the petri dish on which he grew some bacteria, it was contaminated by other microorganisms that secreted some antibiotics that killed these bacteria. And that was the basis, and he understood that there is something that kills bacteria that are secreted from these fungi, and he used this right away not right away, but after a while, for the isolation of penicillin, and that was the first antibiotic to be used, and it was followed by the use of many other antibiotics. So here was again a basic research observation that changed entirely the treatment of bacterial diseases in medicine. Still another example is shown in the next slide. So here you have two investigators, Fisher and Krebs, that discovered phosphorylation, reversible phosphorylation of protein that is very important in signaling, in passing signals from one place in the body to some other cells. And they again wanted to know, they did basic research, but the basic research led to the development of inhibitors of protein kinases, which are now very important agents, especially for the treatment of many cancers. So these are all examples in how basic research at the end led to implications that helped industry and helped humankind. And still another example of the use of the importance of basic research for applications is my own research of how proteins are degraded, on which I am working for quite a while. So I would like to tell you about my work, and I try not to be technical because I understand this is a very intelligent audience, but not necessarily in biomedical sciences. So I try to show my work or the essence of my work in a non-technical language, and then to show how this basic research, which I've been doing for many years, actually at the end helped the treatment of disease. So I'm working on protein degradation. And first, what are proteins? Proteins are the so-called machines of our body. They carry out all the thousands of different processes in our body. They carry out all the chemical processes. There are thousands of different chemical processes, and each process is carried out by a different protein, which are called enzymes. So we need thousands of different enzymes to carry out the thousands of different chemical processes that are essential for our life. Proteins are also carrying out physical processes, such as the contraction of our muscle or the electrical impulse throughout our nerves. So that is also carried out by proteins. Proteins are regulators that regulate a huge number of different processes in our cells, such as cell division. They regulate embryonic development and so on. And another example of the multitude of proteins that we need is the immune response, the different antibodies. We have thousands of different antibodies, each of which is directed toward a different invading agent, because it has to specifically neutralize an invading agent. So there are thousands of different proteins, and we need all of these thousands for our body to work. So how can we have so many proteins? And that is explained by the structure of the proteins. So proteins are built of building blocks called amino acids. Here you see 4 of them. There are 20. So it's just an example. And in the protein, these amino acids are joined like beads, beads on a string. So they are joined together, and usually there are hundreds of amino acids along this chain of one protein. And the difference between the different proteins is the order in which the amino acid building blocks are arranged. So you can imagine that these 20 amino acids can be arranged in millions of different ways, different orders or sequences in a protein that is a couple of hundred of amino acids long. So that is how we can have so many proteins by having them arranged at different orders. And the order is instructed for each protein by its DNA. So DNA is the blueprint that has a certain sequence of chemicals, which sequence actually dictates the ordering of the amino acids in the formation of proteins. And this process of the making of a protein based on the sequence known in its correct sequence, based on the DNA, a chemical sequence of DNA, is called protein synthesis. And it's a fascinating and important process. And because of that, there was a lot of interest 50 years ago when I started to work on proteins, on the process of protein synthesis. But there was much less interest in the opposite process, because proteins are not only assembled from amino acid building blocks, but also disassembled back from proteins to free amino acids by the process of protein degradation. And while there was a lot of interest in protein synthesis, there was little or very close to zero interest in protein degradation in the opposite process. But protein degradation was also important, and that is shown in the next slide. So why do we need protein degradation? One important need is to get rid of abnormal proteins. So proteins, after they are made, they are folded in a certain three-dimensional way, which is important for the function of the protein. But that is a very delicate structure, and proteins get very easily become abnormal by all kinds of damages, such as by oxidation or by other chemical damages, or even just by high temperature. And when they lose their shape, they become abnormal and become toxic. They kill the cells in which they accumulate. So it's important to remove proteins when they become abnormal. So that's one important function of protein degradation, to recognize abnormal proteins and to remove them. But there is another function of just to degrade normal proteins, which have to work for a certain time. And when the time is up, the protein is degraded. So it's like a switch off. So they have a very important switch off regulatory function, the degradation of a protein. That's a way to stop the action of the protein. So protein degradation was known to be important, but it was not known how proteins are degraded. How are cellular proteins degraded at a highly selective and regulated mode? Selective, I mean some proteins are degraded within minutes, and some others are degraded only after many days. So that's selectivity. Each protein is degraded at another rate. And it's also regulated. So some proteins are degraded only when there is a certain signal. How does it all happen? How can the proteins degraded? How can the machinery recognize a certain protein to be degraded at a certain minute out of the many thousands of different proteins that we have? And we found how that happened, and I won't go into the details, but next slide shows you the essence of the system that degrades proteins. So it is by linkage to a small protein called ubiquitin. Ubiquitin is a small protein that gets linked to the protein to be degraded. So, and usually there is a chain of ubiquitins that is made, and that is the signal for the degradation. So there is a marker. So only those proteins that have to be degraded are recognized by certain enzymes that know to find these proteins, and they link ubiquitin to them, and thus they mark them for degradation. And then in the second phase, proteins are linked to ubiquitin are degraded by a complex, by a structure that is called the proteasome. It's like a barrel or a meat grinder into which proteins that are linked to ubiquitin are stuffed inside, and then they're chewed up to their amino acids. So it's like if you want a meat grinder, but a very sophisticated meat grinder, because it chews up or it degrades only proteins that have been first marked by this marking agent. And then ubiquitin is usually released and can be reused again for protein degradation. So this is the essence of the system, and we worked on it for many years. And after we found the basic biochemistry of the system, many other investigators got interested, and it was found that ubiquitin-mediated protein degradation is important in almost every basic cellular process, such as in the control of cell division. So when cells divide, it is by the formation and then the degradation of certain proteins that act at a certain time in cell division. So protein degradation is very important for the control of cell division, which is important for cancer, for example, because in cancer there is an abnormality of cell division. Protein degradation is important in the transduction of certain signals from outside of the cells to inside of the cells. It is important for the regulation of the expression of some genes, such as those involved in inflammation and immunity. Protein degradation is important in the development of the embryo, how an embryo develops from one cell, the fertilized egg, to become a whole organism, the whole body. Protein degradation is important in what is called apoptosis or programmed cell death. And of course, protein quality control is needed to remove abnormal proteins, as I just mentioned. So when I started out, I saw that protein degradation is important, but even I was surprised how important it turned to be in cellular regulation. So protein degradation is important in the health. It's also important in disease. So protein degradation is in abnormalities in protein degradation is important in cancer. Some proteins that are called oncoproteins stimulate cell division, and when they are not degraded, they build up. There's a lot of them, and then there is abnormality in cell division. So a cancer many times is due to abnormalities in the degradation of proteins that control cell division. It's also important in neurodegenerative diseases, such as in Parkinson's or Alzheimer's disease. In diseases, there are some abnormal proteins that accumulate in the certain cells in the brain. They are not removed for some reason, which are being studied. And because they are not removed, they damage these cells and they cause these awful diseases. Protein degradation abnormality is involved in certain mental types of mental retardation, in muscle wasting, and many more other diseases. So protein degradation is now ubiquity-mediated protein degradation that we discovered is now known to be important in both health and in many diseases. And because protein degradation is important in diseases, the pharmaceutical industry got interested, and it is busy to try to find different new drugs that are targeted against the ubiquitin system. And one is shown in the next slide. This drug is called Velcade or bortezomib, and it is an inhibitor of the proteasome, and the proteasome which degrades ubiquitin proteins. And it's very useful against a certain disease, a certain cancer, a bone marrow cancer called multiple myeloma. And for some reason, but it's not completely understood, these cells, cancer cells, are very, very sensitive to partial inhibition of the proteasome, and they go into apoptosis, they die. And because of when they are treated with a low concentration of this proteasome inhibitor. And this treatment, together with some other treatment, completely changed the treatment of multiple myeloma, a disease that was lethal within a very short time, now has been turned to a chronic disease that can afford good quality of life for many years. So it's very satisfying to see that our basic research that we did for many years eventually led to a drug that helps the quality of life of hundreds of thousands of people all around the world. And the drug companies are now busy to try to find other drugs that are targeted against the ubiquitin system for protein degradation. So here you saw how our basic research for many years that we just wanted to know. We wanted to know how proteins are degraded and how is that, how is specific proteins in important processes in the cell are being degraded at the right time. So all this basic research eventually led to application. And the conclusion is shown in the next slide. So basic research is the long-range investment of society, I believe, for technological innovation. If society wants innovation in industry, it has to invest in basic research. And basic research will eventually be translated into great applications. So if we want to have highly original innovation in industry, society should support high quality basic research. So that is more or less my talk, and I wanted to thank the people who helped me for many years. So here are some people in my lab who helped me for many years, like Dvora Ganot, Hannah Heller, Esther Eitan, Sarah Elias, and my wife, Julie Hershko, helped me for a long time. I had many graduate students, among them Aaron Shachanover, who shared with me the Nobel Prize for the discovery, because he worked with me at the time of the discovery, but there were many others who worked on the roles of the system in different processes, and collaborated with good people, including with Irvin Rose, late Irvin Rose from Fox Chase Cancer Center, with whom I collaborated and with whom I was glad to share the Nobel Prize. And here are some recent lab members who work now with me on the role of the ubiquitin system in the control of cell division and mitosis. Thank you very much for your attention.
Thank you, Professor Hershko. It is fascinating to see after 20 years, when 20 years of Nobel Prize ceremony, we have so many applications of the fundamental research you did, and this is, and you mentioned that you have to support the fundamental research, at the same time also the applied research, and this is what SIPS is about. We bring in one roof fundamentalists that deal with fundamental science, with applications, and the industrial scale of production. So that's exactly all you mentioned is SIPS. We bring them together, including mathematicians,
Yes.
including chemists, including physics. As you mentioned, all these are related. And this is exceptional presentation, because it reflects what we are doing. It's a unique conference in the world, multidisciplinary, bringing in one roof all the disciplinaries of science. So thank you very much. It's very, very inspiring.
Thank you.
You wanted to say
Thank you.
something? Because this is
Yeah,
really,
I just wanted to say that again. I am very sorry that I couldn't be physically there in a great meeting, and I talked about medical sciences because that's my field. But I think that the principle of importance of basic research is extending to all different branches of science. So that fits SIPS very well.
yes, we have about three medical symposia. We have oxidative stress, dedicated to Professor Koji Abe. It's the director of a hospital in Japan. We have technologies, innovation in medicine and infectious diseases. So there are three medical symposia with medical doctors. They all appreciate your presentation, but your presentation directs all field of science that SIPS has. So we have 50 symposia with different field of science. So this fits very well. Again, it is my pleasure to congratulate you for the 20th anniversary of Nobel Prize winning in 2004. We are in 2024. So this symposium reflects, this summit reflects that. That's why this is dedicated entirely to you. And now I'm invited you next year, really in person, to come in person next year.
I'll try.
Yeah.
Okay.
So your invitation is public now. So people want to see the real person, the real, behind all this discovery. Okay, then. Thanks a lot.
Good.
Thanks a lot, and we will
Thank
keep in
you.
touch.
Thank you.
Thank you. All the best.
Thank you.